So now what we're going to do is we're going to look at something that's called membrane transport. Um, Basically how cells transport things through their plasma membrane. And this is oftentimes going to involve discussions of different types, a specific category, but different types of integral proteins. So we have two different categories of transport. We have what's called passive transport, and we have what's called active transport. We're going to focus on this one, passive, for right now. Whenever you're talking about passive transport, there's always two things that are going to be required. You're going to need a concentration gradient. So you're going to need a higher concentration of molecules or ions on one side, and you're going to need a lower concentration on the other, right? There's a diffusion gradient. Molecules are going to move from the high concentration to the low concentration. You're always going to get that with passive transport. You need a high concentration on one side of the membrane, you need a low concentration on the other, and those ions or molecules are going to move from the high to the low concentration, and they're also going to do it without energy. They're not going to need energy, typically in the form of ATP, although there are other means of creating energy too that we'll talk about. They're not going to need any energy in order to move from that high to that low concentration. So now what we need to do is look at the different types of passive transport, and then later on we'll come back to the idea of active transport. So here are the different types of passive transport. There's what's called simple diffusion. We're actually then going to talk about diffusion through channels, and then last we have facilitated diffusion. Sometimes you'll see these two, facilitated diffusion and diffusion through channels, lumped together because they're sort of the same thing. And then sometimes you don't. At the end of the day, it really doesn't matter. I'm not going to lump them together. I'm going to differentiate between facilitated diffusion and diffusion through channels for a very specific reason that you'll see. But anyways, we're going to first start off with what's called simple diffusion. This is a type of, all of these are types of passive transport. Molecules are going to move from high to low without energy. All right, so simple diffusion. I can't overemphasize how important this is. Simple diffusion is a type of passive transport. So there's going to be a higher concentration on one side of the membrane and a lower concentration on the other. And the molecule is going to move from high to low without any energy. But what makes this simple diffusion? What actually makes simple diffusion is the fact that these molecules typically do not require any sort of integral protein in order to get through the membrane. They can just rip right through it. How could that be? Well, what you need is for the molecule to be nonpolar. Okay, let me give you an example. A couple. Oxygen, atmospheric oxygen, molecular oxygen, O2 is very, very nonpolar. Carbon dioxide, CO2, is quite nonpolar. It's not perfectly nonpolar. It has some polarity, but it's still very nonpolar compared to other molecules. There is also fatty acids. So you know that fatty acids are made up of hydrocarbons. So like testosterone, estrogen, those are lipid-based hormones. So they're mostly actually made up of hydrocarbons. So lipid-based molecules tend to be pretty nonpolar. Okay, well, what does that actually mean? Let's just pick one of these. We don't need to spend all day going through all the different examples. Let's just pick oxygen. Okay, typically what we see with oxygen is that oxygen is going to be high outside of the cell, and it's going to be lower inside of the cell. Okay? So there's a high concentration in what's called the extracellular or interstitial fluid, and there's a low concentration of oxygen in the cytosol or the intracellular fluid, right? So there's a diffusion gradient. Oxygen wants to move from the high to the low concentration into the cell, and it's going to be able to do it without any energy. So this is a type of passive transport. But the other thing is, since oxygen is nonpolar, the core of the plasma membrane, which is nonpolar because it's made up of hydrocarbons, won't reject oxygen, right? Oxygen is nonpolar. The core of the plasma membrane is nonpolar, so oxygen can just go right through it without needing any integral protein whatsoever. Now think about it. If oxygen was charged for some reason, oxygen would come up to, the core of the plasma membrane, and the core of the plasma membrane would be nonpolar while oxygen would be charged, and then oxygen would get rejected. It wouldn't be able to get through it because oxygen has a charge and the core of the plasma membrane is nonpolar. But oxygen, that's not true. Oxygen is very nonpolar. It's incredibly nonpolar, actually. So the core of the plasma membrane doesn't reject it. Oxygen, being nonpolar, can just go right through it. This is generally true for all nonpolar molecules. Now, there are exceptions to this rule, like large long-chain fatty acids, because of their size, even though they're pretty nonpolar, can still have some trouble getting through the core of the plasma membrane. But generally speaking, if you have nonpolar molecules, they don't get rejected by the core of the plasma membrane because the core of the plasma membrane is nonpolar, and they can travel through what's called simple diffusion. They can move from a high to a low concentration through the core of the plasma membrane without an integral protein. That's going to be true for carbon dioxide, which is typically, let's do that one just real quick. Carbon dioxide tends to be high inside of the cell. Carbon dioxide tends to be low outside of the cell, right? So there's a diffusion gradient there. Carbon dioxide wants to move from a high concentration inside the cell to a low concentration outside of the cell. It doesn't need energy to do so, and it also doesn't need an integral protein to be able to pass through the core of the plasma membrane because, again, carbon dioxide is nonpolar, and so is the core of the plasma membrane. So which molecules can travel through simple diffusion? Typically molecules that are nonpolar, and also very, very small molecules can sometimes sneak their way through the plasma membrane through simple diffusion as well. But that one we won't talk about nearly as much. So what's next on the list, a little bit out of order, but next on the list is what's called diffusion through channels. We're going to look at diffusion through channels next. This is, again, a type of passive transport. All right, so let's talk about diffusion through channels. Diffusion through channels is another type of passive transport. Okay, so you're going to need some molecule that's high in one location and low in another location, and the molecule is going to move from high to low without energy. Okay, so this is another type of passive transport, but why does it fall into the category of diffusion through pores or diffusion through channels, of course, is the question. First of all, the molecule that's going to be traveling is going to be either typically a charged molecule, whether it's an ion or it's polar. Sometimes it's just a very large molecule. This particular molecule, if it's charged, let's say that it's sodium, okay, in this example, right? We know sodium's high on the outside, it's low on the inside, right? So it has a drive to want to come in, right? It wants to move from the outside in, but actually sodium can't, right? Because the core of the plasma membrane is nonpolar. It's made up of hydrocarbons, and sodium is a positively charged ion, so if sodium tries to get past the core, it can't because the core will reject it because it's charged. Okay, well, so if we want to get sodium actually into the cell, what we need is a pore. This is actually a transmembrane integral protein that exists in the plasma membrane. And this pore, what it does is it actually serves as a passageway for the sodium ions to be able to enter into the cell. Right? Sodium can move from a high location outside of the cell to a low concentration inside of the cell, as long as it travels through this pore, this opening that actually exists for sodium ions. Okay, this is true for really all of our cells. All of our cells in our body have some of these sodium pores. It varies from one cell to the next with regard to the number that there are, but there are also pores for potassium. There are pores for chloride. There are pores for calcium. Right? We've got pores for lots of different ions, and the pores are all specific to those particular ions, and they serve as a passageway, a tunnel for those ions to be able to travel through. As long as there's a high location, a high concentration of that particular ion on one side of the cell and a low concentration of that particular ion on the other side of the cell membrane, and there's a pore, then that particular ion will be able to move through that pore from the high to the low concentration. And there's not just pores that exist for ions. There are pores that exist for, particular molecules as well, like pyruvate and all sorts of other molecules as well. Okay? The point is simply this. When you talk about diffusion through channels, what you're talking about is a type of passive transport. A molecule or an ion is going to move from high to low, right, without energy, so it's a type of passive transport. But since the molecule typically is charged, either polar or ionic, it needs a channel or a pore in order for it to actually be able to move past the plasma membrane and move from that one side to the other. So you can easily differentiate between simple diffusion and diffusion through channels, right? With simple diffusion, you still have a molecule like, molecular oxygen, high outside, low inside. So it wants to move from high to low. It doesn't need any energy to do so, and it also doesn't need an integral protein in order to do so because oxygen is nonpolar and the core of the plasma membrane is nonpolar, so it can just go right through it, right? That's simple diffusion. Now with diffusion through channels, again, you have the same thing. You have in this example that I'm giving you, an ion, a sodium ion, that's high on the outside, low on the inside. It wants to come in. It doesn't need any energy to do so. But what it does need is a channel or a pore in order for it to be able to actually get in, because sodium is charged and the core of the plasma membrane is nonpolar. So if you want sodium to come in, you need a passageway for it. Generally, this is going to be true for all charged molecules and ions, and again, this is highly specific to the instance and the cell, but what you'll see is different degrees of these pores for different specific ions and other charged molecules to be able to pass through. Now, what I'll finish off saying is with diffusion through pores, sometimes, the pore is just open all the time. It's just like there are sodium pores embedded in all of our cells, and they are literally just open all the time. There's not very many of them actually in any of our cells, but there's caveats to that. But generally speaking, most of our cells don't have many sodium pores. Okay. And then there are these same sort of integral proteins, transmembrane integral proteins, that serve as pores or passageways for sodium, and they're gated. Okay, there's a gate that exists within them, and that gate has to actually be opened in order for sodium to be able to travel through. Sometimes, it's some intracellular pathway that opens the gate. Sometimes it's a voltage that opens the gate. It's all sorts of different things that can open the gate that we'll talk about throughout the course of this semester. But I just want to be clear that these pores or channels that exist for these different ions, some of them are open all the time, and some of them are gated and have to be opened by some pathway or some mechanism. I'm going to tell you one last thing about these pores, and that's how they actually work. Okay, so these pores are actually made up of a whole, they're an integral protein, and they're made up of all sorts of different subunits. Okay, and the subunits are actually these amino acids that exist typically in a double helix within the different subunits. All right, so how do these pores actually allow for typically charged molecules and ions to be able to pass through them? And the answer, is relatively simple. Okay, so if you take a look, what you'll see is that by being organized into a double helix, the different subunits, the portions, the amino acids that actually face the plasma membrane, those amino acids are nonpolar. So they can exist with the fatty acid tails of the phospholipids, which are also nonpolar hydrocarbons. Okay? And then if you look in the center, kind of the amino acids, in this example would be right here. The amino acids that line the center of the pore, they are polar amino acids, right? They're charged. So why would we want the sort of the opening in the center of the pore to be lined with polar or charged amino acids? And the answer is, well, if we want this potassium ion here, which is charged, to be able to come through the opening in the center of the pore, well, then we need the center of the pore to be lined with amino acids that are polar, right? Potassium is a positively charged ion, and if potassium were to just try to come through the core of the plasma membrane, it wouldn't work because you know the core of the plasma membrane is nonpolar. It's made up of hydrocarbons. It would get rejected. So what we need for the center of the pore to be lined with charged amino acids. And the center of the pore most absolutely is. So that's how these pores are organized. The whole center or lumen of the pore is aligned with charged amino acids, and the periphery, the external periphery of the pores are lined with amino acids that are nonpolar and can coexist with the hydrocarbons, mostly of the phospholipids in the plasma membrane. Now, these different pores, as I've said, and we'll talk about more, are typically highly specific to, they're not always, but oftentimes they're highly specific to a particular ion. And, you know, this particular pore is specific to a potassium ion, and that's also going to be dictated by the different sort of amino acid structure of the pore itself. And some of that, I mean, it's not even fully still understood to this day. Our knowledge of these pores that exist in the plasma membrane really doesn't go much beyond like 60 years. So there are many of these, the different pores are being discovered on a yearly basis. We still are, you know, kind of, this is still in its infancy in terms of discovering all of the different pores and channels that exist in the plasma membrane. So it's difficult to tell you how one specific pore is specific to one particular ion versus another. Some of that's challenging, but the chemistry is certainly there. And really all I'm interested in showing you is how it's possible for these pores to actually allow something that's charged to come in. And that's because the amino acids that line the opening in the center, the lumen of the pore, are charged, just like the ion or molecule that's passing through the pore. Next is the last type of passive transport, and that's called facilitated diffusion. Facilitated diffusion is a lot like diffusion through channels. Okay, what you're going to have is you're going to have a molecule that's high on one side. In this example, we are using glucose. Okay, so glucose is high on the outside, glucose is low on the inside. Great. Okay, so it has a diffusion gradient. Glucose is going to be able to come in and it's not going to need any energy. So this is a type of passive transport, just like diffusion through channels is, just like simple diffusion is. These are all different types of passive transport. But what in particular makes this facilitated diffusion? Okay, you still need a transmembrane integral protein that's going to be specific to the particular molecule. This particular transmembrane integral protein, they're called GLUT or GLUT proteins. They're specific to glucose. Just like the different pores can be specific to different ions, these particular integral proteins that participate in what's called facilitated diffusion are going to be specific to the molecule that they bring in through facilitated diffusion. Okay, so how does facilitated diffusion actually work? Well, again, it's really a lot like diffusion through channels, except it's the way that the integral protein is orientated. So what you'll have is the integral protein will always be open facing either the intra or extracellular fluid and then closed on the opposite end. Let's just focus on this image. What you'll see is this GLUT protein. This GLUT protein, again, this transmembrane integral protein is open facing the extracellular or interstitial fluid, and it's closed facing the intracellular fluid. And then what glucose does, right? Glucose is high on the outside, it's low on the outside, so it wants to come in. And what it will do is it'll come in and it'll dock into this integral protein that is opened up facing the extracellular fluid. Of course, glucose can't just go through the core of the plasma membrane because it's nonpolar, and glucose is polar, it's charged. It's also large. Okay, so glucose comes in and it binds, it docks into this integral protein that's open facing the extracellular fluid. And then once glucose docks inside, what happens is the GLUT protein then closes off facing the extracellular or interstitial fluid and opens up facing the intracellular fluid, and that actually allows glucose to come in. And then after glucose comes in, it reverts back to its original orientation, which is this. Okay, it reverts back to being opened up facing the extracellular fluid and closed facing the intracellular fluid. And then another molecule of glucose can come in, bind to the integral protein, and it can change its orientation to allow the glucose molecule in again. Okay. So when we talk about facilitated diffusion, again, it's a lot like diffusion through channels. It's an integral protein allowing some charged or large molecule to move from a high to a low concentration past the plasma membrane. But when we talk about facilitated diffusion, what it really is, it's how the integral protein operates. The integral protein is always open facing one side and closed facing the other. And then when the actual molecule comes in and docks, it changes its orientation to actually allow the molecule in or to allow the molecule out. So that's really the only way that facilitated diffusion is different than diffusion through channels. It is how the integral protein itself operates. It's the fact that it's open one side and closed off facing the other. With diffusion through channels, the pores are either open all the time or they're gated. They don't function in the same way that the integral proteins that participate in facilitated diffusion do. They're very different. We are going to talk quite a bit about a different, what's called the GLUT4 pathway related to insulin. And you're going to see a lot of facilitated diffusion. And we're going to go through the whole pathway by which insulin stimulates muscle cells to be able to uptake glucose via facilitated diffusion. And then we'll talk about this pathway with relationship to diabetes and exercise and all sorts of interesting stuff. So now you've seen all the different types of passive transport. Passive transport is always a molecule moving from high to low and not requiring energy. If the molecule itself is nonpolar and it doesn't need an integral protein at all, then it can travel through simple diffusion, which is a type of passive transport. If the molecule is charged, either polar or ionic, or sometimes just very large, then it's going to use either diffusion through channels. It's going to travel through a channel that's either open all the time or gated or a combination of the two. Or it's going to travel through facilitated diffusion, where the integral protein is open facing one side, closed off facing the other, and changes its orientation once the molecule docks. Okay, so these are all different types of passive transport that we will come back to throughout the course of the semester. So now we're going to move on to the other broad category, which is active transport. Active transport is the opposite of passive transport. What's going to happen is we're going to move molecules from a low concentration to a high concentration against their concentration gradients. We're going to move them from a low to a high concentration, and in order to do this, energy is going to be required. Sometimes that energy is in the form of ATP, and sometimes the energy is in the form of something else. We're going to look at the first type of active transport, which is called primary active transport. All right, so here's what we know. Sodium, as I've told you, is high on the outside, and sodium is low on the inside. Sometimes what our cells like to do, and I shouldn't say sometimes, they like to do this all the time, is they like to take sodium from a low concentration and kick it out of the cell to a high concentration. That's against sodium's concentration gradient. And let me tell you something else as well. Potassium is high on the inside and potassium is low on the outside. And what our cell does all of the time is it likes to take potassium from the extracellular fluid on the outside and kick it back in, right? So it's moving potassium from a low concentration on the outside to a high concentration on the inside. Okay, so again, this is against its concentration gradient. Cells do this all the time, and in order to be able to move a molecule from a low concentration to a high concentration, energy is going to be required. Okay, that's where the whole idea of active transport comes from. Now, if ATP is the form of energy that's being used, then that type of active transport is called primary active. This image is showing you what's called a sodium-potassium pump. Basically, the sodium-potassium pump works to kick sodium out and kick potassium in. Both of them are being moved against their concentration gradients. We're going to talk about sodium-potassium pumps a lot. And in order to do this, ATP is utilized. ATP comes along, the third phosphate is split off, energy is released, and that allows the integral protein to actually move sodium from a low concentration inside of the cell to a high concentration outside of the cell, and the opposite for potassium. The ATP actually allows the integral protein to take potassium from a low concentration outside of the cell to a high concentration inside of the cell. Okay, so if ATP is utilized by the integral protein to move a particular molecule or ion from a low to a high concentration, then it's called primary active transport. Now, I can just real quickly show you because I was not showing you the actual slide itself. What's happening here in the first image is the sodium ions are coming in, and then ATP, right, is being broken down into ADP, and that's actually allowing for the sodium ions to get kicked out. And again, they're moving from a low concentration inside to a high concentration outside. Okay, and then you can see the same thing for potassium. Potassium is coming into the integral protein, and ultimately potassium is getting kicked in. And again, what we're seeing here is potassium is moving from a low concentration outside to a high concentration inside. And in both cases, this integral protein has utilized the breakdown, the hydrolysis of ATP as the energy that it needs to move these ions from a low to a high concentration. Okay, so that's primary active transport. This is an incredibly detailed step-by-step process by which the sodium-potassium pump works to kick sodium out and move potassium in utilizing ATP. For the sake of what we're talking about right now, I by no means need you to know every single one of these steps, but if you're curious, they are all laid out in this image, because sodium-potassium pumps will come back multiple times throughout the semester in quite a bit of detail. All right, so ATP doesn't always serve as the energy. Sometimes something else serves as the energy. And if it's not ATP serving as the energy, then we refer to it as secondary active transport. That's what I'm showing you here. What we're going to do is we're still going to move a molecule from a low to a high concentration. So we're going to need energy in order to do it. But the energy that's utilized is not ATP. So we're going to call this secondary active transport. Now, I can tell you what's oftentimes utilized for secondary active transport is the diffusion of sodium into the cell. Now, I don't necessarily like how this is orientated, so I'll draw it over for you over here. Okay, so what you'll have is an integral protein like this, and we know that sodium is high on the outside and sodium is low on the inside. Okay, so we know that sodium wants to come in through its natural diffusion gradient, wants to move from high to low. And we also know that as long as there's an integral protein that exists for sodium, sodium will be able to sort of passively diffuse into the cell, through passive transport, as long as there's sort of a pore or a channel for sodium to travel through. And sometimes what happens is sodium will actually diffuse past an integral protein, right? Because it needs to in order to be able to get in. And as it diffuses past that integral protein, the diffusion of a positively charged sodium ion past that integral protein provides that integral protein the energy that it needs to move some other molecule from a low concentration to a high concentration. What I'm showing you is calcium. We know calcium is high on the outside and it's low on the inside. Right, so let's say that we want to kick calcium out. That's actually being depicted over here. Okay, so let's say that we want to kick calcium out. Sometimes, well, first of all, if we want to kick calcium out, we want to move it from a low to a high concentration. We're going to need energy in order to do that. We're going to need some type of active transport. But this time we're not going to use ATP. We're actually going to use the diffusion of a sodium ion into the cell to power the integral protein and give it the energy that it needs to move the calcium ion from a low concentration inside to a high concentration outside. Okay, that is secondary active transport. With secondary active transport, what you do is you use the diffusion of some charged ion or molecule. You're seeing it happen with chloride over here. You use the diffusion of some sort of charged ion or molecule into the cell, and as that charged ion or molecule moves into the cell down its natural diffusion gradient, it moves past an integral protein and gives the integral protein the energy it needs to move some other molecule actually from a low to a high concentration. Right? So you're still getting active transport. A molecule is being moved from low to high, but in order to accomplish this, it's not ATP that's powering the integral protein. It's the diffusion of a charged ion or molecule that powers the integral protein. In advanced physiology, secondary active transporters become very important. I really don't care too much about this terminology, but you're probably going to see it a lot, especially in research articles and in other courses. So I'm going to tell it to you. What you're seeing in all 6 of these is what's called antiport. Okay, so what happens is you have the plasma membrane, you have an integral protein, sodium diffuses in, and the diffusion of sodium in past the integral protein provides the integral protein the energy that it needs to kick some particular molecule out from a low to a high concentration. Okay, if the 2 ions or the ion and the molecule or whatever are moving in opposite directions, as is depicted in every single one of these, then it's called antiport. Okay, this is secondary active transport still, but if the 2 ions, let's just say that they're both ions, if the 2 ions are going in opposite directions here, like if sodium is diffusing in and that provides the integral protein it needs to move calcium from the inside where it's low to the outside where it's high, then sodium and calcium are going in opposite directions, and this is called antiport. This is secondary active transport, specifically antiport. Now, what you'll also find is what's called Symport, and that's where they go in the same direction. Okay, this is the same thing. Over here is secondary active transport. Sodium, we know, is high on the outside, low on the inside, and it diffuses past this integral protein, and it actually provides this integral protein the energy that it needs to move a molecule, in this case glucose, from a low concentration outside of the cell. This happens sometimes, believe it or not, especially within the intestines and sometimes within the nephron, epithelial cells of the nephron. Glucose can actually be low outside of the cell and relatively high inside of the cell. Okay, so sodium diffuses from a high concentration outside of the cell to a low concentration inside of the cell, so it kind of passively moves past the integral protein, and it provides the integral protein the energy that it needs to move glucose from a low concentration outside of the cell to a high concentration inside of the cell. Okay, so this is still secondary active transport. The integral protein is pumping glucose from a low to a high concentration, and it's using the energy from sodium diffusing past it. Now, but if you look closely, what you'll see is that sodium and glucose are both moving in the same direction, right? They're both moving into the cell. Sodium's moving from a high to a low concentration in the cell, and glucose is moving from a low to a high concentration inside of the cell. But they're still both moving in the same direction. So what this will be referred to as is Symport. Okay, this is still secondary active transport, but it's called Symport if the molecules or ions move in the same direction. So now you've seen it. Okay, this is active transport. Active transport is always moving molecules from a low to a high concentration with energy. If the energy that's utilized is ATP, then it's primary active. If the energy that's used is the diffusion of some particular ion, allowing the integral protein to move the molecule from low to high, then it's referred to as secondary active transport. All right, so I have shown you the different types of passive transport and the different types of active transport. And these, again, will keep coming up throughout our conversations.